Our presentation covers the topic of airborne pollen, one of the most widespread allergens worldwide, and why studying it is becoming increasingly important, especially in urban areas. Airborne pollen is produced by trees, grasses, and weeds, with the majority of our study covering the pollen released from trees, and it is a major trigger for seasonal allergies and asthma and other respiratory conditions. While pollen is a natural part of our environment, cities tend to make the problem worse. Urban areas create what is called a pollution-pollen interaction. Vehicle emissions and other pollutants can attach to pollen grains, making them more allergenic and allowing them to stay suspended in the air longer. In addition, the urban heat island effect, where cities are warmer than surrounding rural areas, often causes plants to bloom earlier and for longer periods, making pollen seasons longer and more intense. So why does this matter? First, understanding the pollen concentrations and how they change over time allows public health agencies to issue more accurate allergy forecasts. These forecasts help people with asthma and severe allergies prepare and manage their symptoms better. Second, better pollen data can guide urban planning decisions. For instance, if certain neighborhoods experience consistently high pollen levels, cities can prioritize planting low allergen tree species or adjust landscaping practices to reduce exposure. Third, improved pollen monitoring also has broader implications for climate change research. Changes in flowering and pollen release patterns are strong biological indicators of shifting climate changes. Tracking these trends helps scientists understand how warmer temperatures and increased CO2 levels are influencing ecosystems and, by extension, human health. Looking at the environmental and societal impacts, high pollen exposure can seriously affect quality of life. People miss work, children stay home from school, and productivity drops due to allergy symptoms. Studies have shown that pollen-related respiratory issues contribute significantly to healthcare costs every year. By identifying areas with consistently high pollen concentrations early on, cities can make proactive measures. For example, planting low allergen vegetation, increasing green buffers, or improving air filtration in public spaces can all help reduce exposure. In summary, airborne pollen isn't just a seasonal nuisance, it's a growing environmental and health issue shaped by urbanization and climate change. By studying how pollen levels vary across space and time, we can help public health officials better forecasts, support more informed city planning, and contribute valuable data to climate science. Ultimately, the goal is a healthier, more resilient urban environment, one where both ecosystems and people can thrive despite the challenges pollen proposes. This is where our objective comes in, with our initial hypothesis being, does lowering the relative proportion of male trees in a city result in lower pollen counts and therefore milder irritation? This then pushed us to have an objective of comparing and evaluating the three pollen monitoring instruments, those being the WIBS, Rotarod, and Burkhardt trap, within three equally divided sections of a major city to see how changing the number of male trees affects pollen's negative impacts.
So the first step of this study is to conduct an allergy survey in each zone that we have delineated on the right, zones 1, 2, and 3. The purpose of the allergy survey is to get a baseline of everyone's allergy level before the experiment is conducted. That way we can have a baseline so we can compare it later. We will also conduct a tree survey. We will count how many trees, male, female, and the types of trees in each area, in each zone. And once we have that tree survey, then we can compare it to the allergies and also get a baseline that we can compare post-experiment. We will establish the zones. The zones will be of equal area. They will also have the same amount of trees and the same amount of people. In the timeline of this experiment, it will start in the spring since trees generally pollinate in the spring time, typically between February and April. The first part of the experiment will take place where at the end of the spring, April time, we will conduct the allergy survey, say, hey, what was your allergy level this spring? and then post survey or experiment, we can compare it at the end. So the timeline of the experiment will be 1 year long, and we compare the results at the end of that 1 year. The next step is to delineate these zones. So the zone one will be the control where no trees are touched. All trees are kept the same. People can move in and out of these zones as they wish, but the control is kept the same. Zone two will be the male trees are removed. So we know that only male trees are the ones that pollinate, and that pollen travels to the female trees to create new seeds and fruit and everything. So we will remove only the male trees from this zone, and then from zone three, we will remove all trees to see how it affects the pollen levels. Ideally, we would see zero pollen in zone three. And at the end of the 1 year, we will take a post-experiment allergy survey to see if how the allergies changed from before the experiment to after, before the trees were altered to after. We will conduct a survey gauging what their allergy levels were. The post-experiment tree survey, we will see how many trees were removed and see how the trees affected the stakeholder, which is the population of Columbus. And then we will take those surveys, take also the pollen counts from each zone, and we will analyze the data and get our results.
And to gather the data for our experiment, we'll be using 3 different types of unique equipment. The first of which is the rotorod. Basically how the rotorod works is that it spins 2 rods that are covered with adhesive at a constant rate, kind of sucking up all the particles in the air. And it typically captures molecules in the 5 to 100 micrometer range. The rotorod operates on a 1 minute of sampling for every 10 minutes for 24 hours, which means that the rods are switched out after 24 hours, and typically the results are stained with Kalberla's stain, which turns the pollen grains pink and allows for easy identification under the microscope. And the rotorod is cheap, portable, and its simple design leads to fairly limited maintenance as well as maintenance costs. The next piece of equipment is the wideband integrated bioaerosol sensor, or the WIBS for short. Basically how the WIBS works is that it constantly draws in air. The air is then stuck to an adhesive inside the machine, and typically it can capture molecules between 1 and 30 micrometers. And the WIBS utilizes UV light to excite the biological material in the pollen grains, which turns them fluorescent, which allows for the WIBS to easily distinguish between what is pollen and what is not, and it conveniently does it for you. As well, the WIBS software can automatically track the data, like the pollen shape or the size, which allows for that data to be plotted on graphs, which is nice for the case of the study. And it also enables day-to-day pollen tracking that can be linked to meteorological conditions. So you could look at a specific day with the data and compare it to the weather that day and be like, hey, it was raining on Tuesday and this is what the pollen count is, versus it was dry, really dry on another day and this is how the pollen was affected. So you can use it to make links to the weather as well. But the downside of the WIBS is that it is fairly costly. The third piece of equipment is the Burkhardt trap. And the Burkhardt trap, similar to the WIBS, it continuously sucks in air. This time it's stuck onto a slowly rotating drum with adhesive on it. And the Burkhardt trap typically completes 1 rotation in about 7 days, and then you can then take this tape out, and the tape is cut into 24-hour sections, could be smaller, and then placed onto a microscope slide and then stained, kind of previously to the rotorod results. And this could allow for daily and even hourly tracking of pollen. So you could just cut a 24-hour section into even smaller parts to get more precise data and see how, even over the course of 1 day or 8 hours, if the pollen is going up, down, whatever. And it kind of serves as a middle ground between the WIBS and the rotorod in terms of its cost and complexity.
Okay, so now that we've talked about each of the devices we'll be using individually, it is now necessary to talk about how we're actually going to obtain data from those devices. So for the rotorod, the process for obtaining data is fairly simple. The adhesive from the rods will be removed and a small sample of it will be placed under the microscope. And then you stain it so that it's easier to see underneath the microscope. From there, you count the individual pollen spores that you see. And then this count will be converted to a overall pollen count for the volume that you sampled. This equation is pollen counted times a conversion factor, and then that is divided by the total volume of air that you're sampling. The accuracy of this count is typically dependent on calculating the correct conversion factor, and this conversion factor is based on the magnification you used, and then as a result, the field of view of your microscope. Next, and the more common of these devices is the WIBS, which is the wideband integrated bioaerosol sensor. This automatically outputs numerical pollen counts based on the data it receives. And these measurements are taken continuously so they can be plotted over time, which the software that this sensor operates with automatically plots particle concentrations as well as size distributions over time. So it's easier to see how the data is changing. And in addition, it also outputs other parameters such as the maximum concentration detected, particle count, fluorescence excitation, fluorescence detection, as well as flow rate. All these outputs, as well as the ease of its use, is why it's most commonly used, and it is also used regularly for determining pollen counts around the country. And then the last quantification we have to talk about is for the Burkhardt trap. This process is very similar to the one used for the rotorod. So a portion of the tape that collects the pollen for this trap is placed under a microscope, and this is typically done in 24-hour increments. And what's specifically different about this trap is that a vacuum pump draws in a constant rate of air per unit of time. So for example, a pump could draw in 10 liters of air per minute. And this conversion can then be applied to the overall amount of time to get a volume of air that has been sampled over that time period. And once again, the pollen that you count on your slides that are stained needs to be multiplied by the conversion factor and then divided by the volume of air sample to get your overall pollen count for the volume of air that you're sampling.
The conclusion and implications of this survey, basically, the most important thing is the difference in pollen count, because that is our main study. We need to see how the pollen changed from the beginning of our survey to the end, and that is based on the survey results of the allergies that people experienced. So once we take the survey results, how they differed from the beginning to the end of the 1 year experiment and the difference in pollen count, and we can compare those two sets of data and see if the lower pollen count affected the allergies, if there was a change in pollen count, etc. The stakeholder implications. The stakeholder is the general population of Columbus. We are just trying to see if a difference in pollen count changes the amount of allergies people have and the reactions people have. So if there is less pollen, people tend to be happier because they have less of an allergic reaction to the air. And if there's no difference in pollen, then they will be unchanged. And here are our references.